Okay, so I'm going to be talking about the role of the genes in inflammatory bowel disease. And in general, IBD is really known as a complex disease, which for most patients is secondary to a dysregulated immune response to commensal microbes in the genetically susceptible host. And over the last several years or decades even, there's been a tremendous explosion in our understanding of the different genes and pathways associated with the development of IBD. And many, if not most of them, have to do with the relationship between the host and the microbe. And throughout each of these different discoveries, or on a whole, the question's always asked, so what's the relevance of that specific pathway or that gene to human disease? Although it does seem to make sense, how do we know it's actually a problem? And most of the GWAS identified variants are actually non-coding. So the ones that we are talking about are the ones that actually affect gene expression and then protein expression. Furthermore, most of these genes, or a lot of these pathways, have this fascinating overlap with other immune-mediated diseases, as well as with how our host sees certain pathogens that have been relevant from the beginning of time almost. So you can see that there's sort of this theme where the host is sensing a microbe, and any dysregulation in that pathway can result in an inflammatory response. There's also the issue of a common variant versus a rare variant. So common variants, which are seen in most patients with IBD, might have a small effect, but cumulatively they might result in a phenotype, whether it be mild or severe, versus that rare variant that has a very large effect, hard to find it, but might be the one problem in this patient's disease. So the first time that we actually knew that there was one specific gene associated with IBD, or Crohn's specifically here, was NOD2, a frameshift mutation. And NOD2 is one of the pathogen recognition receptors, so its job is to sense an invading microbe, tell the cytokines to launch an attack, but also responsible for secreting antimicrobial peptides. It's been replicated in different cohorts, pediatric and adult, and there's this phenotype associated where there's this ileal disease, stricturing disease as well, that when combined with other risk alleles can have this more severe prognostic effect, such as surgery. Some of those risk alleles are in other cells that live in the Paneth cells, so that's, or genes. Those are HEG16L1 or IRGM, just a few, and those are obviously important for autophagy, and so it's really that combination of what's going on in the Paneth cell that might cause a subset of patients to have this sort of disease. And over the last several years, there's been a lot of excitement about the Paneth cell itself and how there could be a site of inflammation originating from the innate response. And that's largely because the intestinal epithelial layer itself is our sort of first line of defense. So it's not just a structural defense, but also a functional defense. So in terms of structure, you have the tight junctions, the goblet cells that secrete mucus, which is another line of defense. We have the Paneth cells that secrete antimicrobial peptides, as well as alert the innate and adaptive immune response to start. Additionally, you have cytokines that sort of live really just to focus on the intestinal epithelial layer, and then will alert the rest of the cytokines to launch an attack. Specifically, IL-22, it really functions just on the intestinal epithelial layer, while it's produced predominantly by Th17 in response to what's happening in the intestinal layer, it's been recently discovered the new cell group, if you will, the innate lymphoid cells, and the ICL3 specifically houses these IL-22s to respond to microbes. Over the last 2 years or so, ILC1 and 2 has been found to be incredibly important for the development of older onset or adult IBD, and it's being looked at in early onset IBD as well. In terms of the adaptive immune response, there's obviously all of the pathways seem to be involved, but one of the first ones noted was IL-23 receptor. So the IL-23 receptor had been known to be interesting in addition to the IL-12 and P40 subunit, and what was found that it really was important to sort of launch this T memory cell response and then initiate an IL-17 response. But what's fascinating with some of the discoveries, both in pediatrics and adults, is that there's that protective variant, so there's a decrease in production of CD4 memory cells and CD8 cytotoxic cells. So what was really interesting and cool was that when we understand this very specific pathway can be pathogenic but protective, perhaps we can create a therapy targeted to that pathway and alleviate disease in some patients who have that genetic predisposition. And ustekinumab does exactly that. It really is targeted for the IL-23 receptor pathway. Clearly, it's not going to treat every patient with IBD, and it's not like our silver bullet that we now have, but there are a subset of patients who do respond well to this drug, and perhaps we need to look a little bit more at the patient's individual genomics to know if they'll respond to that drug. A lot of effort and work was done looking at pediatric IBD. Does the pediatric population have the same genetic landscape as adult onset IBD? And for the most part, there's tremendous overlap between the two groups, particularly when we talk about children who are diagnosed about 8 and above. For the children who are diagnosed at a very young age, it's a totally different ballgame, and we'll discuss that in a moment. But for the older children, it seems to be a lot of the same genes. There are a few that are very specific to the pediatric population. This demonstrated that IL-27 was seen mostly in pediatrics. The same paper showed TNF-RSF6B and PSFG1, and another actual 3 or 4 loci within that study. And since then, there are other genes too. But on a whole, kids who are diagnosed between 8 throughout the older ages seem to have a similar background to older onset IBD or adult IBD. So Jocelyn and all published their landmark paper in 2012 that taught us a tremendous amount, or still teaching us a tremendous amount about the pathways involved in IBD for all ages. They demonstrated there's 163 risk loci associated with the disease, 71 of which were pretty new. We didn't really know much about them. But what's really fascinating was besides the tremendous overlap between Crohn's and UC, there is this 71% overlap with other immune-mediated diseases, again making us think that our immune system is really primed to recognize these pathogens that were relevant for centuries, and there's something going wrong with that that's overlapped with all these different processes. So that includes immunodeficiencies, our RA, psoriasis, and spondylosis, ankylosis, but also most fascinating, I think, is for MAP. So there's a tremendous amount of overlap with mycobacterium, which sort of hints to us that, again, there's a central theme of the very, very specific genotype interacting with a microbe, commensal flora or pathogenic flora, resulting in the individual phenotype. So in 2005, Sandy Kim did a beautiful study where she looked at the same genetic background in murine model, so it was IL-10 knockout, and they were exposed to different bacteria or different environment, and they had a very different phenotype. So same gene, different environment, so either germ-free, so they were in a nice environment with nothing going on, and had no inflammation. Those with the commensal flora had a mild colitis throughout, and E. faecalis had severe left-sided disease, while E. coli had a severe right-sided disease. And then she looked at those mice who had a different genetic background with exact same bacteria, and again, you get a different phenotype. So again, it's a very specific genotype microbe interaction. So the role that the actual genes play in each person's disease is probably largely dependent upon how old you are, for the most part, that you're diagnosed. So older children and adults tend to have this polygenic or multigenic disease process. So there's perhaps many different genes of those common genes with that small effect involved in their particular phenotype, and it's a cumulative effect of all these together, versus kids who are diagnosed at a very young age, less than 5, but a very different subset, less than 2, tended to have a Mendelian inheritance or a monogenic inheritance involving genes of the primary immunodeficiency pathways, so not necessarily the same 163. And just to remind everyone, very early onset is an IBD diagnosed in children who are less than 5. They often have this very severe and very different presentation. They're often unresponsive to conventional therapies, and there seems to be an enrichment in rare genes versus these common genes that we just talked about. So while GWAS has been fantastic to identify common variants that might have these small effects, we really can't use that to detect the rare variants. They're minor, or their frequency is just too low, but they might have a very large effect on the individual patient, and we really have to rely on next-generation sequencing to find them. And our hypothesis has been at CHOP, but a lot of centers as well, most centers, is that there are rare and novel variants that are enriched in this patient population involving the immunodeficiency pathways, both with the innate and the adaptive pathways, including a large B cell abnormality as well as this digenic mode of inheritance. So not all of these patients seem to have a Mendelian with one-hit disease, but when you're looking very carefully, you'll start to notice that actually it's maybe 1 or 2 or 3 that come from different parent that are the same pathway converging, and this is why the father or the mother don't have the disease, or they have the disease but not as severe, and the child has a severe presentation. We use whole exome sequencing, mostly a CHOP, as well as a little bit of whole genome. Other centers use predominantly whole genome or targeted genetic approach. So Yuling et al. Published a really beautiful review of the different immunodeficiencies that are associated or seen in very early onset, particularly in the rare genes of common diseases. And it's really, as you'll see, within each single layer of the immune systems and the host defense. So the epithelial layer, as we discussed before, how critical that is. There's a few primary immunodeficiencies that really just are defective in that spot. That includes ADAM17 deficiency, NEMO, familial diarrhea. Any of those defects can really shut down the epithelial barrier when the patient has this sort of rare Mendelian inheritance of those variants. Any step in T and B cell development can occur in VEO and cause severe disease. So for B cell, it can start with development, with migratory, with Wiskott-Aldrich syndrome, with Omenn syndrome, any sort of defect along the CVID pathway or the common variable immunodeficiency pathway can result in VEO. And then for T cells, Tregs are defective secondary to FOXP3 defects, as well as some other defects in that line. And IPEX is a major problem, obviously, for the T cells, including IL-10, that whole cascade, which we'll discuss as well. Phagocyte defects include CGD, or chronic granulomatous disease, as well as leukocyte adhesion deficiency. And again, those severe rare genes are associated with VEO. And then for autoinflammatory, so those are the XIP, mevalonate kinase deficiency, Mediterranean fever, Hermansky-Pudlak, and those can present as a very severe form of VEO as well. So IL-10 was really the first gene that told us this is a totally different ballgame, and that children who are diagnosed under 2 or under 5 are a different disease and a very severe disease. The IL-10 is this heteroteromic complex, so there's 4 subunits, and it's the binding of the IL-10 to the IL-10 receptor that allows for an anti-inflammatory cascade to occur and blocks excessive inflammatory response. Any problem in the IL-10 or the IL-10 receptor alpha or beta will cause really an unchecked inflammatory response to perhaps a commensal microbe and not necessarily a pathogenic microbe. And so these children tend to present within a few months of life with a very severe colonic disease, stricturing or fistulizing disease. There's also a skin disease associated, and they really do not respond to conventional medical therapy or surgical approaches. So if a patient undergoes diversion or colectomy, the disease continues. But what they are cured by is stem cell transplant, and it's sort of critical to understand that this is the pathway involved because there's malignant transformation or potential with IL-10 for different lymphomas. Scott Snapper recently presented his case and published it of a child who was diagnosed with VEO, not sure what was the cause. She failed multiple therapies. She went on to develop lymphoma at around 12 or 13, and she underwent standard protocol for lymphoma, which did not prove to be effective, and was scheduled to get a stem cell transplant, autologous. Luckily, Scott Snapper's group was consulted. They looked for the IL-10 receptor, and instead she got an allogenic transplant from a matched, unrelated donor, which saved her life, because had she gotten her own cells, clearly it wouldn't have turned out well. So chronic granulomatous disease is associated with IBD. It's the rare variants, again, that are seen in kids with the very early onset. So any defect in the complex that allows for NADPH binding production of reactivated species to allow for phagocytosis or killing can result in CGD with multiple abscesses, but also a very severe colonic disease. And Alex Muse and all demonstrated really nicely that the rare variants are associated in the binding of RAC2, which allows for NADPH to then bind to produce oxidative species, but also in other variants that were really common, but the rare form caused this more of a colonic disease. There was really no ileal involvement in these children, no upper tract involvement. They had this very distinct colonic disease that was not responsive to therapy. Knowing someone has CG is critical. You wouldn't want to treat these patients with an anti-TNF because there is potential for sepsis, for overwhelming decreased ability to fight infection. So if you recognize this might be a child who has a CGD pathway, then you would use other therapies in that patient. XIP is another pathway that we see with patients who have VEO. This was first shown in 2011, but subsequently has had multiple cases seen. So XIP's job is to be an antipeptidic protein, so it blocks apoptosis. It also has a critical innate immune response where it works together with NOD2. Any defect in this complex can cause XLP1, which is a triad of excessive susceptibility EBV, HLH, as well as splenomegaly. It's the XLLP2 that we see with patients with VEO. So they have this rare variant of either one or two of these genes that results in a severe colonic disease. And this is just a nice schematic to show what XIP does and if there's some sort of defect, usually a hypomorphic or a null mutation results in VEO, but XIP is necessary for NOD2. NOD2 doesn't really work well without it to recognize an invading pathogen. Same for NOD1 and Dectin-1, which sees fungus. So if XIP is non-functional, there's no clearance of the bacteria or fungi in the innate arm. And then the adaptive arm, XIP is this anti-apoptotic protein, so it blocks caspases from cleaning up extra T cells. Again, so if it's not working, you have both an innate and adaptive destructive process occurring, and you can see why you have this inflammatory response to the bacteria or fungus that are hanging out. And so that causes a TNF response, an increase in the cytokines causing HLH, but also VEO. So CVID has been known to be associated with all ages of IBD, but again, it's this rare variant that causes a severe form in young children. So it's literally any stage throughout B-cell development can result in this unchecked inflammatory response or development. So CD19 was seen with early onset. LRBA was recently published to be associated with children who have very early onset IBD. At CHOP, we have a bunch of patients who, on exome sequencing, we found had defects in this line. So we had CD19, LRBA, as well as an MSH5, which causes class switching. In order to really understand the function of what you find when you do whole exome sequencing, because the variant looks really exciting and potentially cool, but could be totally unimportant, you have to do functional analysis to determine that. So we looked at the memory cells of these patients and saw compared to healthy children or children with VEO without those defects, they had decreased ability to form memory cells as well as plasmablasts. So in general, when you see a patient who has early onset IBD, you still do your standard workup, you still do your labs, infectious rule out, imaging, and upper and lower colonoscopy. But then you really need to look at the immune profile of these children because your therapy will be dependent upon what you find. So you look for their immunoglobulin production, vaccine profile, you look at the T and B cell subsets, as well as CGD analysis with a DHR or NB2. If you have a very specific phenotype and you're pretty sure that you know exactly what you're going for, that it looks very much like one of these pathways, you can do a targeted genomic approach, or you can look for the function of those pathways. You can do an IL-10 receptor analysis or XIP. If you're not sure but you know it's something not good, you can do whole exome sequencing, or you can do a targeted sequencing if you have that profile set up at your institution. Despite the fact that this is clearly, and I'm talking all about this, genomic contribution to the disease, it still requires that interaction with the microbe, with the environment. And so the gut microbiome develops between birth and 3 years of life. And it's probably not coincidental. So every exposure during that time, whether it be mode of delivery, diet, or antibiotics, will shape that microbiome from birth to the adult structure by 3 years of life. So it goes from this chaotic, undiverse sort of environment to the very, very speciated, diverse adult microbiome. So perhaps this is a time period where we can not just learn about the disease in these children, but hopefully at some point maybe intervene earlier on. So in general, it really requires this genomic microbiome interaction at any stage in the immune pathways. And in general, understanding the landscape, the genetic landscape, hopefully will teach us a lot about IBD for all populations, and what you learn about the young children will definitely be helpful for understanding the immune response for the older populations. GWAS was good for some common variants. You'll probably next generation sequencing for those rarer variants. And when you do find something of interest, functional analysis is critical, either through a murine model or translational study, to see how it really is relevant to human disease. And ultimately, I think the goal for all of us is to really individualize therapy to our patients, maybe finding something in those pathways, making us use unconventional therapies that are not really currently part of our arsenal. That's it.
Thank you.